Good morning, students. Today I am here with a new concept, and today I will explain different processes of rDNA technology. So, students, while we are talking about the processes of rDNA technology, so I want to tell you that while we are going to introduce one gene which is of our interest into one host organism to obtain our desirable products, we should have to follow various steps. Now, what are these steps or what are these stages? I am telling you one by one. The first stage is isolation of genetic material. Means in first stage we have to isolate the genetic material of that organism from which we need a desirable gene. Is it clear? Now the second step is uh, cutting of DNA into small fragments. Now we require only a gene, so we have to cut the DNA into smaller fragments. Now what we have to do in the next stage? In the next stage we have to separate out all the fragments of the DNA because we have to select our desirable fragment. Is it clear? Now in the fourth step, what we will do? We will ligate this fragment of the DNA with the vector DNA. Means we will ligate it with the vector or any kind of plasmid. And finally we will introduce this vector into the host cell, and after inserting inside the host cell, the vector will become the part of the host cell DNA, and finally we will go for obtaining product from that particular host. So these are the various steps. First step is isolation of the genetic material, second cutting of the genetic material into various fragments, third we can say that separation and isolation of the DNA fragments, fourth ligation of uh, these fragments into the vector DNA. Now fifth we can say that introduction of or insertion of this vector into the host cell, and final last sixth step I can say that obtaining products from that particular gene of our interest in a well mannered or well developed conditions. So today I will discuss the first three steps. The first step is isolation of genetic material or DNA. Now, as I mentioned earlier that the nucleic acid is the genetic material which is present in all living organisms, and in the most of the living organisms it is present in the form of deoxyribonucleic acid. So we can say that RNA is also a kind of genetic material in some of the organisms, but in most of the cases the genetic material is DNA or deoxyribonucleic acid. Okay, and it is present in pure form, means it is present uh, in pure form. I have to say that it is free from other macromolecules. Okay, and we have to cut it with the help of the a specific restriction enzymes. So this is the later on process I am not going to explain right now. So initially you have to know that the main genetic material is called DNA, and DNA bears various genes, and these genes are basically sequences of nucleotides which are expressing themselves, okay, and helping to produce a specific kind protein or product. So initially I want to tell you that suppose there is a gene of my interest and I want to remove it from that particular organism. Okay, suppose I desire or I will a specific character in a particular organism, and in that particular organism this desirable character is not present, and this character is present in any other organism. So what will I do? I will cut the gene from that one particular organism, and I will introduce the same gene into the that organism in which I require the particular characteristic. Okay, so initially I will go to isolate that particular gene or that particular DNA from the donor cell or the donor organism. So since we know that DNA is not free inside the cell, okay, and it is enclosed within the membranes. So in order to release DNA along with the other macromolecules such as proteins, polysaccharides and lipids, bacterial cells, plants and animal cells are treated with the various kinds of enzymes. Now I want to tell you that once again I am telling you that suppose I want a gene, and a gene is present in a either in a bacterial cell or in a plant cell or in any kind of fungal cell या animal cell. ठीक है, तो इनिशियली वट जीन को बाहर निकालने से पहले इनिशियली मैं पूरा का पूरा डीएनए बाहर निकालना पड़ेगा ना मुझे. तो वट विल आई डू? तो इनिशियली if I want to I want to isolate a pure form of DNA, तो मुझे क्या करना होगा? मुझे उस सेल के अंदर मौजूद जो आपके लिपिड्स हैं, जो आपके पॉलीसेकेराइड्स हैं, जो प्रोटीन्स हैं, उनसे फ्री करके डीएनए को बाहर निकालना पड़ेगा। ठीक है, तो for this process, if I want to isolate DNA from one bacterial cell, so I have to use one enzyme, lysozyme. Similarly, if I want to remove the DNA from a plant cell, then I have to use cellulase enzyme. If I want to release or remove this particular DNA from a fungal cell, then I have to use chitinase enzyme. So different kinds of enzymes are used to isolate DNA from different kinds of living organisms. Then after, since we know that along with it other macromolecules are also present, so as well as some other other nucleic acids are also present, RNA is also present. So to remove RNA, I have to use or I have to treat this DNA with ribonuclease enzyme, because this enzyme will digest RNA completely. Similarly, if I want to remove proteins from that particular DNA, so then I have to use protease enzyme, and this protease enzyme will digest proteins from that particular DNA. ठीक है, तो I told you that while we are going for the isolation of the DNA, if we are isolating DNA from bacterial cell, we have to use lysozyme. ठीक है, if we are isolating DNA from plant cell, we have to use cellulase. If we are removing DNA from the fungal cell, we have to use chitinase. For removal of RNA, I have to use RNase, सॉरी, या RNase का जो फुल फॉर्म होता है उसको बोलते हैं राइबोन्यूक्लियेज. ठीक है, similarly if I want to remove proteins, then I will use protease. ठीक है, now finally what can I do now? Other molecules, I will remove some other molecules. ठीक है, now I have removed proteins, I have removed RNA, I have removed the cell wall and other contents. Suppose there are some other contents like carbohydrates and lipids and some other kind of contents, so I can use different techniques or different appropriate techniques to remove such kind of impurities from the DNA. And finally I will go for the precipitation of the DNA. Now I will use chilled ethanol, and with the help of the chilled ethanol, while I will pour this chilled ethanol with the help of the wall of the vessel, so I will observe that the DNA threads are suspended over the uh, that particular solution in which the DNA, in which the cell is kept. ठीक है, तो finally with the help of the ethanol we can precipitate out the DNA threads or DNA segments. ठीक है, now DNA is completely isolated, the first step is completed. Now I have isolated the DNA fragment. Now अब दूसरा स्टेप जो है, I have to follow the second step, and the second step is called cutting of DNA at a specific location. अब मुझे पूरे DNA से क्या चाहिए? मुझे तो अपने इंटरेस्ट का जीन चाहिए खाली. Now I will, तो अपने इंटरेस्ट का जीन मुझे कहाँ मिलेगा? DNA का एक छोटा सा टुकड़ा मेरा इंटरेस्ट का जीन होगा. तो now what will I do? I will cut the DNA into various fragments. Now you know very well that if I want to cut DNA into various fragments, so what will I use? So I have to use restriction enzyme, or basically restriction endonuclease enzyme. ठीक है, now restriction enzymes are used for the digestion uh, of the DNA, or digestion means to cut down it into the various fragments. ठीक है, the restriction enzyme digestions are performed by incubating purified DNA molecules with the restriction enzymes. This is done with the optimal condition for the specific enzyme. So as you know that a specific restriction enzyme is used to cut DNA into various fragments, and for it the DNA is I have to say that the DNA is incubated with the restriction enzyme, and optimal conditions are kept in which the DNA breaks down or cuts down into various fragments. ठीक है, now DNA is now cutted down into the fragments. Now I am coming to the last and very important step. Now I have to separate out or I have to isolate DNA fragments, because I need a single fragment which have my interest of gene. Now the cutting of DNA by restriction endonucleases results the formation of the various fragments of the DNA, now which can be separated by a technique, and the technique is called as gel electrophoresis. Now these segments of the DNA can be separated out with the help of a technique, and the technique is called as gel electrophoresis. Now what is gel electrophoresis? I am showing you. Since the DNA fragments are negatively charged, ठीक है, DNA is negatively charged molecule, we know very well that. ठीक है, so that's why these can be separated by applying an strong electric field. ठीक है, विच मेक्स डीएनए मॉलिक्यूल टू मूव टुवर्ड्स द एनोड. तो इफ़ वी विल अप्लाई इलेक्ट्रिक फील्ड, ठीक है, देन व्हाट विल हैपन, सिंस डीएनए इज नेगेटिवली चार्ज, तो inside the electric field the DNA which is negatively charged definitely it will run towards the positive charge or anode. ठीक है, तो under an electric field, ठीक है, तो we can separate out the DNA fragments by the process of gel electrophoresis. So in this process the gel or the matrix is used as एगारोज जेल. तो यहाँ पर मैट्रिक्स के तौर पे एगारोज जेल का यूज़ किया जाता है, ठीक है, एंड दिस इज एक्सट्रैक्टेड फ्रॉम अ काइंड ऑफ सी वीड. ओके, now DNA fragments are then released into the agarose gel, and then after that two electrodes are kept into that agarose gel and electric field is then applied. So definitely one happens when electric field is applied, तो agarose gel creates sieving effect. ठीक है, the smaller piece can move more faster in that particular gel uh, in comparison of the larger pieces. तो कहने का मतलब ये है कि जो छोटे पीस होंगे फटाफट जेल के बीच से निकलते हुए एनोड की ओर ज़्यादा दूर तक पहुंच जाएंगे, और जो बड़े पीसेस हैं वो दूर रह जाएंगे. ठीक है, तो hence the smaller fragments, the smaller the fragment size, farther it moves. The separated DNA fragments can be visualized only after the staining the DNA with the ethidium bromide followed to the exposure of the UV radiation. Now इसके बाद आपने क्या करना है, now आपने एगारो जेल में इनका सेपरेशन कर लिया. Now you have smaller segments near to the anode and the larger segments farther from the anode. Now you have to stain this agarose gel, or you have to stain these DNA fragments with a special dye, and the dye is called ethidium bromide. Name of the dye is ethidium bromide. Now after it you will pass it under the ultraviolet radiation. While you will pass it under the ultraviolet radiation, so you will observe a orange or reddish color, bright orange colored bands of the DNA. ठीक है, and now after that what can you do after staining, you can cut down these bends from the agarose gel, and you will remove the DNA fragments from this gel, and the process is called as elution. ठीक है, now they will be settled down into the gel. After staining they have color, you can easily identify them, and then after you will cut or you will remove them from the gel, and the process of removing of the DNA fragments from the gel is called as elution. ठीक है, thus the DNA fragments are purified and used for the construction of the recombinant DNA by joining with the cloning vectors. Now we have a DNA fragment of our interest, means I mean to say that now we have a DNA fragment which have gene of our interest. Now we can ligate this fragment with the vector in the next step. I think so it's enough for today. Tomorrow I will discuss what are the next 3 events of genetical engineering or processing of rDNA technology. Till then, thank you, bye bye, and have a nice day.